Physiology Section 3. In this first chapter, we will cover excitation, contraction, coupling. We will start first with some basic histology of skeletal muscle structure. Skeletal muscle is composed of many myofibrils, as illustrated on this slide. From Z-band to Z-band represents the sarcomere. The sarcomere is important because it is the functional unit, the basic functional unit of skeletal muscle. So you can think of the sarcomere as the basic functional aspect of skeletal muscle. Here's another picture of a sarcomere, actually a couple of sarcomeres here, and they are labeled for us. Again, from Z-band to Z-band represents the sarcomere. Now, when skeletal muscle contracts, the A-band, which is the myosin, notice the nomenclature here. We have myosin filaments, actin filaments, and titin. And the A-band represents myosin, although there is some actin interspersed within there. But the A-band is basically myosin. And the myosin is not going to change in length when the muscle contracts. However, the I-band, which is titin and actin, will shorten, and also the H-zone will shorten some as those actin molecules, those actin proteins, move into them. So the A-band doesn't change length, but the I-band and the H-zone do. Titin is very important here because it anchors myosin. So effectively, it is an anchoring protein that connects myosin to the Z-band. We'll talk about some pathology of titin in just a bit. All right, again, a little more of the structure. The T-tubules are extensions of the surface membrane. So illustrated on the slide, you can see, or in the figure on the slide, you can see the T-tubules. And basically what they are the surface membrane diving down inside the muscle fiber down to the myofibrils. It is an extension of the surface membrane. And remember, in skeletal muscle, the name of the surface membrane is the sarcolemma. The terminal cisternae is part of the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium, and most of the calcium is stored in these terminal cisternae. So the terminal cisternae is where we're going to find lots and lots of calcium. Again, the SR has a lot of calcium. Now, for the structural proteins, the proteins of the thin filament. Actin is a structural protein, thin filament again, and it will have binding sites for myosin. So the actin-myosin interaction will be the key for generating active force, as we will discuss in more details in just a moment. So that's the basis, or the basics of the actin molecule. Tropomyosin is also a protein. It blocks myosin binding sites on actin. So it will prevent the interaction of actin and myosin. The next protein is troponin. And technically, troponin is composed of three different subunits. Troponin T, which will bind to tropomyosin. Troponin I, which binds to actin and inhibits the contraction. And troponin C, which is a calcium binding component of the tropomyosin protein. So again, the major proteins that we have involved here, two not listed, titin and myosin, but the ones of the thin filaments are actin, tropomyosin, and troponin. So these are the key proteins involved in the function of skeletal muscle and things to look for on USMLE Step 1. All right, if you look at our resting muscle, again, here are our proteins, actin, illustrated kind of by the circles, the troponin-tropomyosin complex by this thin bead that's kind of woven through, There's our troponin with its calcium binding site, and there's myosin, which is the cross bridge. That's the myosin head that is labeled, and the underneath part is the myosin tail. Notice that actin and myosin are not interacting when we're in resting muscle. There is no binding of actin and myosin. Now, when calcium goes up, it's going to bind to troponin C, which is going to roll the tropomyosin molecule out of the way and expose the myosin binding sites on actin. And that's what's illustrated in the figure that just came in. So again, myosin has binding sites on the actin molecule, but they are covered by tropomyosin in the resting state. And what's going to uncover that will be a rise in cytosolic calcium. So once again, under resting conditions, we have a low cytosolic calcium, and so there's very little calcium to bind to troponin, and therefore actin and myosin cannot interact. When calcium binds to troponin C, the troponin-tropomyosin complex moves away, exposing the binding sites on actin for myosin, which we illustrated on the previous slide. Myosin has an ATPase, and the splitting of ATP puts myosin into a very high energy state. So think of it as a stored energy state. The other thing it does is when the myosin has split ATP into ADP and phosphate, this puts myosin in a very high affinity for actin. So in the resting state, myosin ATPase has split ATP into ADP and phosphate. That's got myosin in a high energy, high affinity state. Once the myosin binds to the actin, we're in this high energy state. This chemical energy coming from the splitting of ATP is transferred to mechanical energy, which causes myosin to pull on the actin filament. This generates active tension and is referred to as the power stroke. So once actin and myosin bind, that high energy state is transferred to the actin molecule, and we are pulling on the actin protein. Now, for a couple of types of contractions we should appreciate for muscle physiology, and thus for USMLE Step 1. If the force generated by the muscle during the power stroke is sufficient to move the load, then the muscle shortens, and this is termed an isotonic contraction. Contrast that with an isometric contraction. If the force generated by the muscle is not sufficient to move the load, then the muscle doesn't shorten. And we will bring this back in again when we talk about mechanics of muscle in the next chapter. All right, let's walk through a single cross-bridge cycling event. Here we are in the resting state. Actin and myosin are not interacting, they're not binding, but myosin has split ATP. So myosin, remember, when it splits ATP, it's in a high energy, high affinity state. High affinity for the actin molecule. It really, really wants to bind to actin, but it can't because tropomyosin is covering the binding site on actin for the myosin protein. Again, tropomyosin is interspersed with troponin, and we have low calcium, so tropomyosin is covering the binding site. Once cytosolic calcium rises, will bind to the troponin C. This will roll the tropomyosin-troponin complex out of the way. Tropomyosin will no longer cover the binding site on actin for myosin. Once that binding site is exposed, step 2, actin and myosin bind together. Again, remember, myosin was in a high energy, high affinity state. So once that binding site is exposed, it will immediately bind the actin molecule. Once myosin binds, it was in a high energy state, so we convert that energy to a mechanical pull. Effectively, myosin will pull the actin, and this will produce active tension. That is our power stroke. Now the next step is ATP. ATP will come in and dissociate actin and myosin. So one of the roles of ATP here is to bind to the myosin. Once it's bound to actin, it will bind the myosin and split. It will dissociate actin and myosin. And it will put myosin into a low energy, low affinity state. So one of the roles of ATP, dissociate actin and myosin. The other role that we've already mentioned is it puts myosin in a high energy state, and that ATP provides the energy for the power stroke. Now let's think about, when do you not have ATP in your cell? Oh, well, the only time that occurs that you don't have ATP is when you're dead. And that is the basis of rigor mortis. Actin and myosin bind together. There's no ATP to dissociate them after you're dead, and that generates the stiffness of rigor mortis. So that's a single crossbred cycling interaction power stroke. Again, step 1, resting. 2, actin and myosin combine because cytosolic calcium went up. 3, we convert that chemical energy into mechanical energy. Myosin pulls the actin, that's the power stroke. Then ATP comes in and dissociates actin and myosin and puts myosin in a low energy, low affinity state. But don't forget, myosin has an ATPase, so after that dissociation, the myosin ATPase will split the ATP, putting myosin back into a high energy and high affinity for actin state. And if cytosolic calcium is high, then again actin and myosin will bind, and this crossbred cycling will just continue as long as cytosolic calcium is elevated. Alright, now that we've discussed the proteins and some basic structure and these proteins and how they interact, we mentioned several times how important the cytosolic calcium was to the process. In the resting state, cytosolic calcium is low, actin and myosin cannot bind and interact, no power stroke. But again, sarcoplasmic reticulum has a very, very high concentration of calcium. There's a huge, huge electrochemical gradient for calcium to diffuse from the SR into the cytosol. Now, this calcium coming in again initiates active tension, the power stroke. So let's discuss in detail the regulation of cytosolic calcium. We're going to have 2 key receptors involved here. The first is called dihydropyridine, which we'll abbreviate as DHP. And the second is the ryanodine receptor. These are going to be our 2 key receptors. We'll start first with dihydropyridine. Technically, dihydropyridine is an L-type calcium channel. It's a voltage-gated calcium channel that's located on the sarcolemmal membrane. However, it's kind of a misnomer to consider it a calcium channel in skeletal muscle. Why is that? And that is because calcium does not flux through this receptor in skeletal muscle. Okay, so again, the sarcolemma has DHP, but calcium doesn't flux. So effectively, there are no calcium channels on the sarcolemma, even though DHP normally acts like one. It's different in skeletal muscle. It behaves as a voltage sensor, not as a channel through which calcium fluxes. And that is a big point for skeletal muscle. The second receptor is the ryanodine receptor. This is a calcium channel on the SR membrane. And we'll look at a picture of both of these on the next slide. In resting muscle, ryanodine is blocked by DHP. This prevents calcium from diffusing from the SR into the cytosol. And again, on the next slide, we will look at these in a pictorial format. All right, here's a picture showing the regulation of calcium. Here we are in the resting muscle. I have highlighted DHP and ryanodine. Again, DHP is a voltage sensor that is associated with the sarcolemma membrane. So it's on the sarcolemma. It's a voltage sensor. Notice that ryanodine is my calcium channel on the SR. Now, in the resting state, notice that DHP is blocking the ryanodine. The best way to think about it is think of DHP as like a cork in a wine bottle or a champagne bottle. The champagne bottle is just filled with all this calcium. Think of champagne as calcium. But it can't come out because of the cork. In skeletal muscle, DHP serves as the cork, and the neck of the bottle you can think of as the ryanodine receptor. So at rest, we have the champagne corked, none comes out. Now, when an action potential hits the sarcolemmal membrane, so we now have a skeletal muscle action potential, notice how it dives down into the T-tubule. Well, the action potential is a rapid depolarization. It is a big voltage change. DHP is behaving as a voltage sensor. So in response to this action potential, DHP undergoes a conformational shift, as they would say in biochemistry, causes a conformational shift in the DHP so that it no longer blocks the ryanodine channel. So you can think of it as the action potential is you popping the cork on a champagne bottle. Once we pop that cork, we have a huge electrochemical gradient, so calcium diffuses into the cytosol. And again, this rise in calcium binds troponin, and that's what initiates cross-spread cycling and the power stroke. The other aspect that calcium does, as I've highlighted on the slide, this calcium rise in the cytosol will also activate ryanodine channels and open them. This is referred to as calcium-induced calcium release. So the rise in cytosolic calcium will open other ryanodine channels, and we'll get a greater flux of calcium into the cytosol to initiate cross-spread cycling and, again, active force. This cross-spread cycling just continues and continues and continues until cytosolic calcium is lowered. So we have to get the cytosolic calcium back down in order to prevent actin and myosin from interacting. So in other words, relax the muscle. Well, relaxation, therefore, is mediated by pumping calcium back into the SR, and that is mediated by a calcium ATPase that we refer to as SERCA. So here's our regulation of cytosolic calcium. On the left is the resting state, low cytosolic calcium. DHP is a cork in our champagne bottle. So calcium or champagne can't come out. The action potential causes DHP, the voltage sensor, to uncork, and calcium comes flooding out. This also opens more ryanodine channels, calcium-induced calcium release. We relax the muscle by pumping the calcium back into the sarcoplasmic reticulum. This requires ATP, and the pump that does that is called SERCA. All right, so let's recap what we just stated, and we'll just put it in a text format. The skeletal muscle action potential gets initiated at the neuromuscular junction. Hopefully you remember the neurotransmitter that's released. If you said acetylcholine, very good. Do you recall the receptor it binds to? Yes, it's the nicotinic muscle receptor, nicotinic, which is a ligand-gated ion channel. And when acetylcholine binds, we will see a change in the conductance of both sodium and potassium, but sodium is a key player. That's what initiates depolarization. We then get to threshold in the skeletal muscle membrane, and there's our action potential. This action potential travels down the T-tubule, and this voltage change causes this conformational shift in DHP. Again, DHP is behaving as a voltage sensor. This pops the cork on our champagne bottle. In other words, we remove the block on ryanodine, and the champagne can come flowing out. The champagne is calcium. All it has to do is follow its normal concentration or electrochemical gradient, and we get a flood of calcium coming out. This calcium opens more ryanodine channels. This is called calcium-induced calcium release. This rise in cytosolic calcium binds to troponin C. That moves tropomyosin out of the way of the myosin binding site on actin, and this creates the power stroke, which is active tension. The calcium is pumped back into the SR by a calcium ATPase, which is technically called sarcoplasmic endoplasmic reticulum calcium ATPase. Not a fun word to say. I'm very much prefer just to say the word circa. Circa is so much easier to say. So this is going to cause relaxation. Circa causes relaxation. Because this fall in cytosolic calcium, now tropomyosin can once again cover the actin's binding site for myosin, and the muscle relaxes. Bottom line, fundamental, circa causes relaxation. All right, a recap of the key points. Contraction, relaxation states are determined by the cytosolic levels of calcium. The source of calcium, all calcium, big point in skeletal muscle, all calcium comes from the SR. And this calcium is going to bind troponin C, and that's what moves tropomyosin out of the way, no longer blocking actin's binding site for myosin. Two ATPases are involved here. The myosin ATPase, which again puts myosin in a high energy, high affinity state. And also circa requires ATP because it pumps calcium back into the SR, and that's what terminates the contraction. So it's all related to that cytosolic calcium. Also, not on the slide, but remember, another role of ATP is to dissociate actin and myosin. All right, can you remember the important pathology related to this process? Can we remember malignant hyperthermia? Malignant hyperthermia is a genetic alteration in the ryanodine channel, which makes it susceptible to certain compounds like succinylcholine or the inhalation anesthetics. When a person who has this genetic alteration is exposed to these compounds, then the ryanodine channel opens up and calcium comes flooding out. Well, crossbred cycling generates heat, and that's the reason that the patient rapidly heats up, hence the term malignant hyperthermia. I bet you can remember what you treated with as well, can't you? Did I hear? Yes, I heard you say it. Dantrolene. Very good, dantrolene. All right, this slide just kind of lines everything up for us. At the bottom, we have membrane potential. That's the skeletal muscle membrane. Next, above that is intracellular free calcium. Next is active force. As we indicated, we get an action potential. This will cause the release of calcium, as illustrated on the slide. This rise in calcium will evoke muscle contraction. That's active tension due to crossbred cycling. That is a single twitch. Now, it's very important to notice that the membrane potential in the skeletal muscle fiber has completely repolarized well before we even see the active force. So, the skeletal muscle membrane is ready to fire another action potential before we actually see the active force. And that becomes important because there's two ways that skeletal muscle can increase its force. One will be summation. Summation is one. The second is recruitment. Now, for skeletal muscle, and again I want to emphasize this is skeletal muscle, you release enough calcium from the SR to saturate all the troponin C binding sites, which means we're going to bring in all the myofibrils, all the sarcomeres are involved. Okay, all available cross bridges are activated. So I can't enhance force by increasing cytosolic calcium because I already release enough with a normal twitch to saturate all the binding sites. So we have to increase force two different ways, summation and recruitment. Summation. Summation means we fire multiple action potentials prior to force development. Again, remember that membrane completely repolarized before we saw the force. Each action potential will cause a pulse of calcium, and you'll see a picture of summation in just a moment. Each pulse of calcium initiates cross-bridge cycling, and because the muscle has not had time to relax, or we get another pulse before we even hit the peak tension, the mechanical force adds on to the force from the previous action potential. So this is what we mean by summate. It adds on. And this summation can continue until the muscle tetanizes. Tetany is when free cytosolic calcium remains high, so cross-bridge cycling is just continuous. It just never stops. As long as we have high calcium, it's binding to troponin, tropomyosin's out of the way, actin and myosin can interact. And that is tetanus. Let me show you a figure of that on the next slide. So the upper picture shows membrane potential for the skeletal muscle. The lower shows the active force. Notice we fire an action potential, we get a twitch. Fire another action potential, we get a twitch. Now, if we start increasing the frequency of those action potentials, the force begins to add on because the muscle hasn't completely relaxed. That is summation. All the way up until we get full-blown tetanus, which is continuous cross-bridge cycling because cytosolic calcium is staying high. So one way to increase force in skeletal muscle is to summate. The second way to increase force is recruitment. Now, by definition, an alpha motor neuron innervates multiple muscle fibers. And the alpha motor neuron and all the fibers that it innervates is called a motor unit. So if I recruit more motor units, which means I just bring in or I start activating more motor units, this engages more muscle fibers and in turn causes greater force production. So hopefully recruitment makes good sense. We're just bringing in more and more muscle fibers, and the more muscle fibers I engage, the greater amount of force. So two ways to increase force in skeletal muscle: summation and recruitment. All right, skeletal muscle is a striated muscle, as is cardiac muscle. And we're going to have a lot of similarities, but we'll also have some differences. Let's just look at the idea of the similarities between skeletal muscle and cardiac muscle. They both have the same functional proteins: actin, tropomyosin, troponin, myosin, and titin. And remember, titin provides that elasticity of the muscle. And dysfunction in titin has been shown to be related to the cardiomyopathies, both dilated and restrictive cardiomyopathies. A rise in cytosolic calcium initiates cross-bridge cycling, which is exactly what we would predict. Since they both have troponin and tropomyosin, ATP plays the same role in both. It puts myosin in a high-energy, high-affinity state, and we need it to convert that chemical energy into mechanical energy. We also need ATP to dissociate actin and myosin. We also need ATP to power the circa pump for relaxation. Okay, both have circa. Both also have ryanodine receptors on the SR, and they show calcium-induced calcium release. Now, the big difference here, though, is the source of calcium, and we'll talk about that in just a moment. All right, differences. Skeletal muscle. All calcium comes from the SR. Not true in cardiac muscle. Extracellular calcium does flux through the L-type calcium channel, so it comes from extracellular into the cytosol. So extracellular calcium is involved in contraction of cardiac tissue, but not skeletal muscle. Now, in cardiac tissue, the magnitude of calcium release can be altered, calcium release from the SR. You don't saturate all the troponin binding sites, so you don't get all the fibers engaged. So if we increase calcium release, we will increase force of contraction. That, of course, is contractility, also known as the inotropic state. We talk more about contractility in the inotropic state in section 4. But this is a big point regarding cardiac muscle. Cardiac cells are electrically coupled by gap junctions. These do not exist in skeletal muscle. For this reason, cardiac muscle acts as what we call a functional syncytium. This just means they're all engaged, so we get one unit contraction of the heart, which is very beneficial because we're trying to eject blood. Another big difference is on cardiac myocytes. Cardiac myocytes have a sodium-calcium exchanger. They do have circa, as we mentioned before, as does skeletal muscle, but cardiac tissue also has a sodium-calcium exchanger on the sarcolemmal membrane. 3 sodium in, 1 calcium out. A sodium-calcium exchanger on the sarcolemmal membrane. Now this becomes very important for USMLE Step 1. And the reason for that is this is the reason that digoxin increases contractility. Remember, digoxin inhibits the sodium-potassium ATPase. This tends to increase sodium inside the cell, and by increasing sodium in the cytosol, we reduce the activity of this sodium-calcium exchanger. And again, remember, this sodium-calcium exchanger is pumping calcium out of the cytosol into the interstitial space. Well, if I reduce the activity of that sodium-calcium exchanger, more calcium gets taken up by the SR, which means I can release more calcium on subsequent beats. And by releasing more calcium, I increase the force of contraction, and again, we refer to that as contractility. And this is therefore the mechanism by which digoxin is able to increase the contractile state, or the inotropic state. That's another word for contractility. Again, we mention that frequently in section 4 of this book. All right, the very last aspect of this book is to look at the action potential for cardiac cells versus the muscle twitch. And notice something. Cardiac muscle is still in absolute refractory when the muscle twitch force is beginning to fall. Now remember, in skeletal muscle, we repolarized before the twitch even started. And because of that, we could summate the force. Well, because of this very long plateau in a cardiac action potential, the muscle twitch is already starting to relax when the cardiac cell is still in absolute refractory. And as you recall, absolute refractory means you can't get another action potential. Now, because we're not getting action potentials, multiple action potentials before the twitch, then we can't summate. So in other words, because of this long absolute refractory period, the twitch tension already starting to fall, cardiac muscle cannot summate. And if you can't summate, you can't go into tetany. And this is the mechanism by which cardiac muscle cannot tetanize. Which is a good thing, because we really don't want cardiac muscle to tetanize, now do we? I mean, tetany is this sustained type of contraction, and we don't want that in the heart. Even though it's more force, right? Tetanic contraction is more force in skeletal muscle, because that's maximal summation. There is more force associated with tetany, but it's not good for the heart, because the heart needs to relax in order to refill. So this is very beneficial from a functional perspective. All right, that's excitation contraction coupling for striated muscle. We really focused in mostly on skeletal muscle, but we did bring in the other striated muscle, cardiac. Next up, we'll talk about muscle mechanics, skeletal muscle mechanics.